Good day everyone. Today we will be presenting the article Synthesizing Life by Jack W. Szostak, David P. Bartel, and P. Luigi Luisi. This article focuses on an important question in biochemistry and biology. Can life be created from simple chemical components in the laboratory? Our presentation will explain what the article is about, why it matters, what the authors proposed, and what is still unknown. For today's discussion, we will cover the following parts. First is the objectives and goals of our presentation, an overview of the article, its impact and legacy in science, what the authors proposed, what the article does not claim and what remains unknown, and lastly, how the article fits into broader origin of life and synthetic biology research. Our presentation has four main goals. First, we want you to understand why this article is important, especially its impact and scientific significance. Second, we aim to explain what the article proposed, particularly the idea of creating lifelike systems artificially. Third, we want to clarify what the article does not claim and the questions that are still unanswered even today. And lastly, we want to show how the article Synthesizing Life fits into broader research about the origin of life and synthetic biology. So what is the article Synthesizing Life by Jack W. Szostak, David P. Bartel, and P. Luigi Luisi is about? The paper was published in Nature in 2001. In it, the authors argue that it is scientifically plausible and useful to aim for creating very simple cells called protocells in the lab. Minimal systems that combine two essential components of life. The article Synthesizing Life is a scientific idea that asks, can we build the simplest form of life in the laboratory? So the authors Szostak, Bartel, and Luisi suggest that we can try to create very basic protocells. And these protocells are like extremely simple versions of living cells. The goal is not to copy modern life, but to understand how life might have started from simple chemicals. So by building these minimal systems, scientists hope to learn what the essential ingredients of life truly are. The authors explain that life needs two main parts working together. First is an informational subsystem or a genetic material. Example of this is the RNA, which is capable of heredity, replication, and mutation. So an informational subsystem, this is like the instructions for life. In early life, this could have been RNA, and it should be able to store information, copy itself, and change over time. And second is a compartment or a membrane. Example of this is a simple lipid membrane that can enclose the informational material and separate inside from outside and maintain a controlled environment. So a compartment or a membrane acts like a container. It separates the inside of the protocell from the outside. This is important because life needs a controlled space where reactions can happen. And together, these two components form the basic foundation for a simple living system. So the article's impact and legacy. The article is regarded as a landmark foundation of bottom-up synthetic biology, pushing the goal of building artificial cells from simple chemical components. It showed that life might emerge from minimal features, a basic membrane, a replicating informational molecule, and compartmentalization, rather than complex modern cell structures. So this article became highly influential because it clearly laid out a realistic way to build artificial cells from simple materials. It helped scientists focus on the minimum traits needed for life. A simple membrane, a molecule that can replicate, and a protected internal environment. So the paper showed that life does not need modern complexity to begin. just a few key features can already produce lifelike behavior. Next is, it helped drive extensive protocell research, including work on lipid vesicles, self-assembling membranes, encapsulated ribozymes, minimal genetic systems, and compartmentalized biochemical reactions. It also influenced major debates on defining life, especially the idea that life could be viewed as a self-sustaining chemical system capable of Darwinian evolution. So the ideas in the article encouraged a lot of research on protocells. Scientists started exploring how membranes can form naturally, how RNA can function inside those membranes, and how small chemical systems can grow or divide. It also influenced discussions on one big question: what is life? The article highlighted that life may simply be a system that can replicate and evolve, even if it is not as complex as modern organisms. According to the article, the authors proposed that primitive membranes could form from simple fatty acid lipids, which naturally self-assemble into vesicles under the right conditions. And inside these vesicles, short RNA strands, including catalytic ribozymes, could replicate or drive basic chemical reactions. So the authors proposed that early cell membranes could form by themselves from simple fatty acids. These molecules naturally gather into bubble-like structures when placed in water. Inside these membrane bubbles, short RNA molecules could perform useful tasks like helping chemical reactions happen or even copying themselves. So this combination of a simple membrane plus functional RNA could be a realistic starting point for the first protocells on early Earth.
Moreover, the model combines membrane first and genetic system first ideas, proposing that both must arise together to form a functional protocell. Their model suggests that neither the membrane nor the RNA alone is enough to form life. Both have to be present and working together. And lastly, the article asks, how simple can a cell be and still count as a living? Identifying the minimum features needed to shift from chemistry to life. The article leads to an important question: what is simply a system that we can still call living? The authors argue that once a system can keep itself organized, store information, and replicate, it starts to bridge the gap between non-living chemistry and living biology. The authors emphasize that the lab-built protocells may not reflect the actual process that occurred on early Earth, so experimental success does not guarantee historical accuracy. The authors remind us that even if scientists build protocells in the lab, this may not be the same what happened during the origin of life. Experiments shows what is possible but not necessarily what actually occurred billions of years ago, and they also note that then and now no fully functional synthetic cell capable of self-replication, metabolism, sustained growth, and evolution has been achieved. This means that no has created a fully independent synthetic cell yet, one that can grow, divide, and evolve all by itself. And lastly, because there is no universal definition of life, determining the exact point at which a protocell become truly alive remains difficult. Meaning, since scientists still debate the definition of life, it is hard to pinpoint exactly when a protocell becomes truly alive. How synthesizing life fits into broader origin of life synthetic biology research. The article fits into bottom-up synthetic biology, which builds lifelike systems from simple chemical components rather than stripping down existing organisms. Explanation: the authors support what we call bottom-up synthetic biology. This means building lifelike systems from simplest materials instead of modifying existing organisms. By studying minimal systems, scientists can identify which features are absolutely necessary for life. It also helps them understand how life might begin naturally from simple components. It aligns with minimal life and protocell research, which examines how basic chemical assemblies can show lifelike traits such as compartmentalization, replication, heredity, and primitive metabolism. The protocell model is very important in origin of life studies. It helps researchers imagine how early Earth might have produced the first life structures. By recreating simple membranes, primitive genetic replication, and basic metabolic reactions, scientists can test hypotheses about how life might have first emerged without relying on modern cellular machinery. As knowledge of probiotic and systems chemistry grows, the papers of framework remains influential, guiding studies on how life might have originated on Earth or on other worlds. Even after many years, the article is still widely used in research. As scientists learn more about chemistry and how molecules behave, the protocell framework remains a powerful model. It helps researchers explore not only how life started on Earth, but also how life might appear on other planets with different environments.